All right, AMP1 students, in this video we're going to finish up talking about the last few learning objectives for Chapter 12. So I'm going to click to our learning objectives. So in the last videos we talked about neurophysiologies. We ended talking about action potentials and how they're propagated and generated. So the next couple of objectives is actually a review from Chapter 10. So we have already talked about events that occur at a chemical synapse when we talked about the events at a neuromuscular junction in Chapter 10. And we also talked about the role of calcium and the release of neurotransmitters. So I will just briefly show you, we'll go back to the textbook. They do talk about what's happening at the axon terminals or synaptic bulbs, which we call the transmissive segment of a neuron. And this kind of shows the axon terminal and the steps that we talked about when we talked about the events at a neuromuscular junction for Chapter 10. So that's a little bit of a review, but realize that you need to still understand what's going on at a chemical synapse. Okay, and know the role of calcium as well in that process, which we've talked about before. All right, so the next thing that we're going to cover is these last few objectives. So we should know the similarities between graded potentials and action potentials that occur along neurons, and we should also know their differences too. So the textbook actually has a table which shows kind of all the differences between graded potentials and action potentials. So one of the big things to remember is where they happen. So the graded potentials, and remember when we say graded potential and action potential, we're talking about that change in the membrane potential away from resting membrane potential. So that change in the electrical charge across the plasma membrane. So graded potentials only happen along the dendrites and cell body because that's the only place where you can find chemically gated channels. So, and then action potential is a little bit different. They only happen along the axon of neurons, and that's because along the axon is where you find the voltage gated channels for like sodium and potassium. And then the direction of the voltage change, so whether it becomes more positive or goes more negative. When it comes to a graded potential, it can go either or. So it could become more positive, the resting membrane potential, and that's depolarization. Or it could become more negative. So that's what we call hyperpolarization. So either one of those could happen. It kind of depends on if we want to excite the postsynaptic neuron. So that's when we'll get a positive change in the voltage or the membrane potential. And then, or if we want to inhibit the postsynaptic neuron, then we'll get a inhibitory. We call it an inhibitory postsynaptic potential, and they call this an excitatory postsynaptic potential. So it could go either way. When it comes to an action potential, both things happen during that action potential. So you will get a positive change in the voltage, so it will become more positive, but then it will then go back towards negative or back towards resting membrane potential, and then actually go more negative than resting membrane potential. So it actually goes positive, then goes back negative, and then it goes back to resting. The amount of voltage change, it's pretty small if you compare the change that you get when the action potential is happening along the axon. So small change in voltage if we're talking about graded potentials, large change if we're talking about action potentials. And then the degree of voltage change, it's dependent on the magnitude of the stimulus. So if we have a lot of neurotransmitters that are releasing from presynaptic neurons and binding to a lot of the sodium ion channels, chemically gated sodium ion channels, then we have a large stimulus. Then we can get a change in voltage that's much larger. So, but when it comes to an action potential, it's kind of all or none. So we get the change in the voltage So it starts at negative 70 for most neurons, goes up to positive 30, comes back down to about negative 90, and then goes back to negative 70. So it doesn't vary. It always happens in that particular range for the membrane potential. So it's kind of an all or none phenomenon that you get with the action potential. So you don't have like a weak action potential, or you don't have like a real strong one. It's just always the same. As long as you reach threshold, you change that membrane potential where voltage-gated sodium channels will open, then you get a action potential where the membrane potential changes to positive 30 and then repolarizes back down to negative 90 and then settles back on negative 70. All right, duration, it's kind of more limited for the graded potentials, but when it comes to an action potential, it can vary depending on the length of the axon. So it'll vary. When it comes to graded potentials, it's pretty standard, so just 1 to 2 or 3 milliseconds. And then the distance traveled, so the dendrites in the cell body, that part of the neuron where graded potentials happen, they're not really long, so that's a shorter distance that the change in membrane potential has to travel. When it comes to an axon, they're much longer. They're the longest process of our neurons. So the action potential is going to always travel a much longer distance than any graded potential along the dendrites in cell body, just because the axon is so much longer than the dendrites in cell body. And then the change intensity. So this is where the graded part comes for graded potentials. So where the sodium or whatever ion is changing the membrane potential. So if sodium's coming in, if sodium's coming in, then that flow of sodium will change as far as how far it will flow across that dendrite and cell body. So the intensity of that sodium flowing will decrease. When it comes to the action potential, the voltage-gated channels get opened, and all the sodium, comes in and flows in the same amount each time. So the intensity is the same throughout that process. So those are our similarities and differences between graded potentials and action potentials that you should be familiar with. Okay, and we do have a few questions on the velocity or speed of an action potential down an axon. And so we are going to look at those factors. So there's 2 of them. Let's see if I can get that in there together. Yeah. So the speed or velocity of an action potential down an axon, there's 2 things that can change that speed. So one is the diameter of the axon. So if we have a larger diameter axon, a bigger diameter, so we have some neurons that are bigger than other neurons, then we will be able to get a faster speed of the action potential down that axon. And that's because we have less resistance. So remember we talked about resistance is that opposes the flow of charged particles. So if we have that larger diameter, there's less resistance for the flow of those charged particles. So they can happen much more quickly, moving, and that'll cause the action potential to go down that axon much more quickly than a smaller diameter axon. So that's one factor. And the other factor is myelination, which we've talked a lot about in this chapter. And that's kind of the role of myelin. If we have an axon with myelin around it, the action potentials actually happen at each node of Ranvier or neurofibril node. So they kind of leap down the axon, and so that makes the speed of the action potential much faster. So if we have myelinated axons, our speed of action potentials are faster. If we have unmyelinated, the speed of those action potentials are slower. All right, so those are our 2 factors that will determine the speed of our action potential. And then the next objective is how action potentials vary in frequency. So let's head to that part of our textbook. So this area is talking about the frequency of action potentials. So remember we talked about how action potentials are all or none events that occur along a neuron. So they either happen and they happen in the same amplitude all the way down the axon, or they don't happen. So we don't have like weak action potentials and we don't have like strong action potentials. So sometimes our brain and spinal cord kind of need to know how intense a sensation is. For example, like a really bright light is more intense than a normal amount of light. A loud noise is going to be more intense than a more normal level noise. So in order for our neurons are still sending action potentials that are exactly the same to the brain and spinal cord when we have these sensations of light or sound or touch of various things. So sometimes it would be hard for the brain and spinal cord to know like how intense those sensations are, because the action potentials are exactly the same they either come in or they don't come in. So how the central nervous system can kind of determine if like maybe a sensation's more intense is because of the frequency of the action potential. So if a bright light is shined in our eye, we will have more action potentials, more frequent ones, running along our optic nerve to our occipital lobe of our brain. And so then our brain will be like, oh, we're getting like very frequent action potentials. So the brain interprets that as a very bright light, as opposed to a more normal light. You turn on the light in a room. That's also going to send action potentials, but not as frequently as with a really bright light. So the brain and the spinal cord can kind of integrate, kind of know how intense that sensation is in that way. All right, so that is our frequency of action potentials. And then the last two of our learning objectives are a repeat of something that we learned in Chapter 10. So remember, we talked up here about the events at a chemical synapse. Remember, acetylcholine was the primary neurotransmitter for the stimulation of skeletal muscle cells to contract. That was the neurotransmitter that was the primary one. So describe how acetylcholine functions as a neurotransmitter, and describe the three different mechanisms for removing the neurotransmitter from the synaptic cleft. Hit the wrong button there. Let's see what page there we go. Let's see if that's on the next page or we have to go back. So this goes over more, here we go. So this goes over, and again, this is sort of a review, just like our events of the chemical synapse are a review. So when it comes to describe how acetylcholine functions as a neurotransmitter, it's released from the little synaptic vesicles of an axon terminal of a motor neuron generally. Could be other neurons too, though. So, and then it will diffuse across the synapse and bind to its acetylcholine receptors. So that is how acetylcholine functions. And then there are ways that we are able to remove acetylcholine from that synapse. Okay, so I found it up here. We have the removal from the synaptic cleft, the acetylcholine. So one of the ways, so it's down there in the text, but after acetylcholine is released from that by exocytosis from that axon terminal, and we no longer need acetylcholine to bind to these receptors because we don't have that signal coming anymore. Then there is an enzyme that is produced. It's called acetylcholine esterase. So hopefully that looks familiar to you. And that will come in and it will take acetylcholine and break it into acetic acid and choline, or acetate and choline. And so those two molecules will either diffuse away from the synaptic cleft, or choline's a lot of times taken up by the axon terminal, so we can like recycle it as well. So that's one way for removal of acetylcholine from the synapse. The other way is it diffuses away and gets taken up by glial cells, so that's why it's out of there. And then there are some neurotransmitters that can be taken up by the